So hello friends, so in this video I am going to discuss how this use of alcohol that is damage the liver and cause alcoholic cirrhosis. First thing. Second thing I want to discuss that is what are the structures found in the histophysiology of liver. And the third thing I want to discuss you about the histopathology of liver. That means what are the structures found in the histopathology of liver. So that's all that I am discussing in this today's video. So please stay tuned. So friends, so for understanding that is how this excess alcohol that is damage the liver. So before knowing this, you should know that a little bit about alcoholic metabolism. So see that alcohol in our liver is converted into acetaldehyde by the enzyme alcohol dehydrogenase, and this acetaldehyde is converted into acetate by the enzyme acetaldehyde dehydrogenase, and this acetate convert into CO2 and H2O. Now here that is one thing yes that you should remember is that the amount of this alcoholic dehydrogenase is not limited in our body, it is abundant, but the amount of this enzyme acetaldehyde dehydrogenase is limited in our body. So see that, and that portion I have written that in our body the amount of alcohol dehydrogenase is not limited, whereas the amount of acetaldehyde dehydrogenase is limited. And this is the most basic and the important part that you should remember. Now please see this equation. So here that is when you take excess alcohol, so out of that 100% of the excess alcohol taken, all the 100% alcohol converted into acetaldehyde by the enzyme alcoholic dehydrogenase. So therefore that is 100% acetaldehyde is formed. But out of that 100% acetaldehyde, all percentage of acetaldehyde is not converted into acetate, as acetaldehyde dehydrogenase is unable to convert all the acetaldehyde into acetate. So out of the 100%, so let's that is 60% of acetaldehyde convert into acetate, and then so here CO2 and water are formed. But the remaining 40%, see that remaining 40% of acetaldehyde so here accumulated in liver, and that accumulation of that is remaining acetaldehyde in our liver so here damages the cells of liver, that is hepatocytes, and leads to that is pathological condition known as alcoholic liver disease. So in this way that is excess alcohol uses so here damages the liver and cause this liver cirrhosis. So friends, now I am going to explain what are the structures found in the histophysiology of liver. Now, so for understanding the histophysiology of liver, so please see this diagram. So by seeing this diagram, so you can relate that in the histophysiology of liver, all the hepatocytes, they are arranged in the form of lobules, and that lobule is the hepatic lobule. So you can see that inside each lobule that is hepatocytes are found, and these hepatocytes they are arranged in a form of cords. So the first thing that you should remember is that in the histophysiology of liver that is hepatocytes are found and the cords of hepatocytes are found. That means they are arranged like a rows. So cords of hepatocytes are formed, and these hepatocytes they are arranged in the form of hepatic lobules. So hepatic lobulated pattern is also found. Now see that, and in this histophysiology of liver you can notice that so here central vein and here portal tride are also found. Now again see this diagram. So by seeing this diagram, so you can relate what are the structures found in the portal tride of a liver in a normal situation, and that structures are portal vein, hepatic artery, and hepatic duct or bile duct. So these three structures are found in the portal tride. Now again see both the diagrams together. So by seeing the diagram, so you can relate that in a normal situation that in the portal tride nor in the space between the cords of hepatocytes in any region that is no neutrophils nor any macrophages are found. So you should also remember that in a normal situation, no neutrophils are found in the portal tract, nor any neutrophils found in the space between the cords of hepatocytes. So these all are the here normal findings in the histophysiology of liver. Friends, about the histophysiology of liver, what I have written. So the first thing, that is no neutrophils found in the portal tract. In the portal tract, only hepatic artery, hepatic duct and portal vein are found. So this is the first finding. And the second is that hepatic lobulated pattern is found, and this hepatic lobulated pattern is found due to arrangement of hepatocytes. And third, that is the cords of hepatocytes are found. And see that normally between the space between the hepatocytes, no neutrophils are found. So between the space of hepatocytes, no neutrophils are found. So these three are the here normal findings of histophysiology of liver. So friends, so you should know that there are three stages of alcoholic liver disease. Now here alcoholic liver disease, that means that is the liver damage or that is a destruction of liver caused due to the prolonged use of alcohol. So this leads to alcoholic liver disease. Now here you should know that in the alcoholic liver disease there are three stages. The first one is the fatty liver stage of alcoholic liver disease, or this is also known as alcoholic steatosis of alcoholic liver disease. So this is the first stage. And the second stage is the alcoholic hepatitis of alcoholic liver disease. And the third stage is the alcoholic cirrhosis of alcoholic liver disease. So these three are the stages of alcoholic liver disease, and I'm going to discuss the histopathology of each of these structures one by one. So let's begin. So friends, so this is the fatty liver stage of alcoholic liver disease, or you can say alcoholic steatosis stage of alcoholic liver disease. Now, and this is the histopathological structure of that fatty liver stage of alcoholic liver disease. Now I am going to tell you that is what are the structures found in this histopathological structure. So first of all, see that in this structure, that is the central vein is not affected, so this is normal, and this is the portal tract. So as you already know that in the portal tract, that is the hepatic artery, portal vein and bile duct are found, and all these things are also normal. So central vein and portal tract, they are not affected in this fatty liver stage, whereas all the here that is the hepatocytes which are found between the central vein and the portal tract, they are affected. Now see there, so the first structure that is found in this histopathological feature of this fatty liver stage is the microvesicle. Now here, what is microvesicle? So see there in the hepatocytes, so normally that is the nucleus and cytoplasm are found, but in this fatty liver stage, that is the excess fat molecules, so here deposited in this hepatocyte, and then this excess fat molecules deposited in the form of vacuoles, and this abnormal deposition of fat molecules in the form of vacuoles in this hepatocyte, so here that is leads to the formation of a abnormal structure known as microvesicle. And you can see that in this microvesicle, that is the structure of hepatocyte is like a flower. So this is the main, that is the microvesicle. So in this microvesicle, so that is the fat molecules accumulated in the form of vacuoles. Now, so the first one, microvesicle, is discussed. Now the next one is about a macrovesicle. So see that in the macrovesicle, you see that all the microvesicles or all the fat molecules which are deposited in the hepatocyte in the form of vacuoles, all those, so here that is the fat molecules that is the combined together, and this combination, so here leads to formation of a large vacuole, and then this large vacuole, so here that is shifts the nucleus from center to the periphery. So this is the macrovesicle. So what is macrovesicle? So in the macrovesicle, all that is fat molecules in the form of vacuoles combined together to form a one large vacuole, and this large vacuole deposits the nucleus towards the periphery, and this structure leads to the formation of macrovesicle, and in this macrovesicle the nucleus is shifted towards the periphery. So this is the second abnormal structure. Now the third abnormal structure is the formation of fatty cyst or the fat cyst, when the fat molecules burst, or the hepatocytes containing these fat molecules burst, then this fat cyst formation is done. So these are the main abnormal structures so here found in the histopathology of the fatty liver stage of alcoholic liver disease. So see, that is what I have written. So here is a microscopic features of alcoholic steatosis stage of alcoholic liver disease. The first one is that is a microvesicle droplets of fat appear in the cytoplasm of hepatocytes. And how these microvesicle droplets of fat appear? That due to the, so here that is a deposition of excess fat molecules in the cytoplasm. And second, so here formation of macrovesicles, and these macrovesicles formed when microvesicle droplets of fat combine together, and in macrovesicles the nucleus so here displaced towards the periphery. And the third is the, so here that is a fat cyst formation is done. So these are the, so here that is abnormal, so here that is a microscopic features of that is the fatty liver stage of alcoholic liver disease. So that's all about the first stage. So now I'm going to discuss to you the, so here second stage of alcoholic liver disease, that is the alcoholic hepatitis of alcoholic liver disease. Now so see that, so you can see that, so here these are the some abnormal structures in the hepatocytes, and these are, that is the ballooning degeneration. And here what is ballooning degeneration? So here that is a ballooning degeneration occurs when the hepatocytes that is undergo swelling due to excess fat accumulation, and this ballooning degeneration also occurs due to necrosis, and also so they undergo swelling, and as a result that is they become swell like structures like balloons. So therefore that is a ballooning degenerations. So the ballooning degeneration is the first finding, and the second finding is that is Mallory's Hiling Dank body. And here see them in some of the hepatocytes, so here that is the excess pinkish eosinophilic, that is the inclusion, so here that is the materials deposited, and this excess pinkish eosinophilic inclusion bodies in the cytoplasm of hepatocytes, and these are known as Mallory's Hiling Dank body. So what are Mallory's Hiling Dank body? So these are the, so here that is the pinkish eosinophilic inclusion material, so here deposited in the cytoplasm of the hepatocyte, and these are known as Mallory's Hiling Dank bodies. And also see that, so this is a portal tract, as so here that is the hepatic artery, bile duct and portal vein are found. And see that in this portal tract also, so here neutrophils are found, so neutrophilic infiltration is found in the portal tract. And also in the spaces, so here between the hepatocytes also, that is a neutrophilic infiltrate is also found. So these are all the abnormal findings. So once again I'm going to tell. So the first one is a ballooning degeneration found, so this is a ballooning degeneration, and the second one is a Mallory's Hiling Dank body, and the third one is a neutrophilic infiltrate found in the portal tract and also in the spaces between the hepatocytes. So these are all the abnormal findings. So see that what I have written. So the microscopic features of this alcoholic hepatitis of alcoholic liver disease. So the first one is a ballooning degeneration of hepatocytes occurs, and why this occurs? Because of the excess fat deposition, and this leads to the swelling of the hepatocytes, so here just like balloons. So therefore that is a ballooning degeneration hepatocytes occurs. And the second one is the Mallory's Hiling Dank body seen, and these are eosinophilic intracytoplasmic inclusion, so here seen in the perinuclear location. Perinuclear location, that means outside the location, so outside the nucleus. So you can see that this pinkish intracytoplasmic inclusions, they are not seen inside the nucleus, they are seen outside the nucleus, so therefore perinuclear. And a third one is a neutrophilic infiltrate seen in portal tract and in the space of Disce. So these are all the, so here that is the histopathological structures of alcoholic hepatitis or alcoholic liver disease. So now this is the third stage of the alcoholic liver disease, that is the alcoholic cirrhosis. So the alcoholic cirrhosis, and these are the, so here that is a histopathological structures. So here you can see that in these structures, that is all the hepatocytes, they are not arranged in the form of rows. So here just like in the previous one, so here in this case, that is the hepatocytes, they are, so here that is converted into nodules and they are arranged in the form of nodules first here. And you can see that, so here between any two nodules, so here that is a fibrous septa is found, and this fibrous septa is also formed due to the, so here that is the myofibroblast proliferation, and this myofibroblast is formed when the erythrocells, they are mutated and they are converted into, so here that is the myofibroblast, and this myofibroblast, so here leads to excess, so here collagen fibers deposition, so therefore that is the fibrous septa is also found. So fibrous septa is also found between the, so here nodules, and here see that in this third stage, that is no central vein, no portal tract is found. Only, so here that is the excess number of, that is hepatic or bile ducts are found, and then this, so here that is excess and the proliferation of the bile ducts, so here leads to ductural proliferation. So what are abnormal findings? So see that the nodular pattern is found first thing, and in this nodular pattern, so see that all the, so hepatocytes, they are similar to each other, they have similar structure, they are looking similar to each other, so therefore uniform size micronodules, also nodular pattern, and also fibrous septa is also found between any two micronodules, and also see that the excess number of hepatic or bile ducts are found, and this is known as structural proliferation. So see that, so these are the microscopic features. So the first one is a nodular pattern, so uniformly sized micronodules are present, and second, between any two nodules fibrous septa seen. The third, that is a ductural proliferation seen, and this is found due to the excessive proliferation of bile ducts. And fourth, that is a no portal vein seen. So these are all the abnormal histopathological structures.